Hi everyone. I'm going to speak to you today about the International Mouse Phenotyping Consortium and how this is a large-scale effort to uncover gene function to inform human health. I'm a coordinator of mouse informatics at EBI. We're a team of 10 bioinformaticians, biologists, and software engineers, and most of them work on this project in varying capacities. The IMPC website is at mousephenotype.org. Feel free to look along. So the IMPC is a consortia of the world's leading mouse research centers. You can see the various members on this map here. And together we're working towards some common goals. The biggest being to create and characterize 20,000 knockout mouse strains or lines, and to try and do this by 2021. This would be one knockout strain for every protein coding gene that's a non-olfactory receptor gene. We have already produced about 5,000 knockout lines, and we're looking to produce 5,000 to 7,000 more in the next 5 years. This project's funded by various national funding agencies. The biggest is the Knockout Mouse Program Common Fund from the NIH, which funds mouse production phenotyping in North America and the UK, and also funds all the informatics, as well as other national funding from England, Germany, France, and many other nations. All strains produced by the IMPC consortia are publicly available through repositories, and we try and publish and annotate data in near real time, all free to use. So the origins of the IMPC came from a meeting, Banbury meeting, in Cold Spring Harbor in 2003. And at the time, the mouse genomes had just been sequenced and published in 2002. And so some of the world's leading researchers got together to describe how we could use this resource and how we could translate it to human health, as the human genome had just been published recently too. Such illustrious people as Francis Collins and Chris Austin were at this meeting. So what they concluded is that we could build a library of embryonic stem cells where each colony would have a vector that would knock out the function of that gene. And so from the spring, the International Knockout Mouse Consortium, which ran for 10 years, and this was a project that was funded in both the United States and Europe, and the consortia working together to generate an embryonic stem cell library for trying to get every protein coding gene. They got up to over 18,000 colonies. This wind down a couple of years ago as the IMPC started ramping up. And the IMPC is its goal is to use these embryonic stem cells to create knockout mouse strains and to do a broad-based phenotyping. And then, as you can see in this little pyramid, this figure comes right out of that meeting in 2003. It was envisioned that then you could use these mice and data to do more in-depth phenotyping, and that is happening currently. I'll touch upon that at the end of my talk, looking at immune, embryo, bone, and aging aspects. So the knockout alleles used by the IMPC that were created by our predecessor IKMC project come in two different flavors. There's the KOMP Volcogene, which is a vector that ablates the whole protein coding sequence of a gene and replaces it with a LAC-C reporter and a Phlox neomycin selection cassette. Then there's the UKOM alleles, which is a little more complicated, where a critical exon, in this case exon 2, is floxed, and upstream of that is a gene trap vector that is LAC-C and a neomycin that has a stop codon here. So you have the strong splice acceptor. So in many cases, this will prevent the function of the gene. Now, in the presence of Cre recombinase, you can remove this critical exon and make a true null, where the gene lacks a critical exon. But the strength of this allele is you can also use a flip recombinase to remove the lac and neomycin and have a conditional ready allele, so that you can knock out this gene and with a Cre strain that expresses it just in selective tissues. This Team 1A alleles proved to be very popular with other researchers. So of course, since the IKMC finished this project, CRISPR technology has come along. And the consortia is now adopting that method because it's so much cheaper and faster to produce mice. And so right now, the centers are ramping up their efforts to do simple deletions of critical exons, and you can see the various alleles that are generated there. And also, there's some R&D trying to create conditional alleles, since researchers really like that. This is not in a large-scale effort, but because there's such a large consortia, there are a number of strains being made to try and find ways to do LOXP insertion in an efficient manner. So once the mice are produced, we want to characterize them. And so the IMPC has worked together to develop a standardized phenotyping pipeline that's captured in our EMPRESS resource. Starting at 9 weeks of age, mice are assessed in a series of tests that measure different physiological aspects of the mouse. Each test is designed so it doesn't impact in subsequent tests, and because of this, we're not able to do certain things like immunological challenges, since this would alter downstream tests. When the mice have reached 16 weeks of age, they're sacrificed and given an extensive necropsy so we can learn as much as possible at this mouse and gather as much data so that we can get this data to the public and hopefully allow research to decide if this knockout strain is worth pursuing. Now, going into this project, we knew that some of the mice would be embryonic lethal. And so the NIH said we should try and get as much data from these embryonic lethal mice as possible. So we've added an embryonic phenotyping pipeline where mice are assessed that are in the breeding scheme when there's no homozygote mice detected. Researchers at the individual centers will do a gross morphology check on the embryo around embryonic 12.5 to see characterize what developmental disorders can be observed grossly. And then based on that, we'll then do an extensive image analysis at an earlier or later time point. And this is now being done on approximately a third of the lines that are embryonic lethal. And then in addition to this, in most cases, the phenotyping centers will take a heterozygote mouse, so a mouse with just one knockout allele from these embryonic lethal lines and characterize them as an adult so we can get some hypomorphic information. So there is over 10 centers now that are characterizing these knockout mice and putting them into the phenotyping pipeline, both embryonic and adult. And all this data is being centralized with the MPI-2 consortia. So the MPI-2 consortia consists of MRC Harwell, which is based near Oxford, where they have a team of biologists and developers who have developed a resource that allows upload of data and quality control checking with feeding back to the information to make sure we get the best data out of the centers. At EBI, we have a tracking resource that tracks all the production of the mice. And once this data has been approved by trained data wranglers, it's released to archive based at EBI, where analysis is done, phenotype calls are made, and the data is pushed out to the public. So I mentioned the data wranglers, and they are a critical part of this process. And so here we have a screen capture of the PhenoDCC that is used to communicate between the data wranglers and the individual centers. In this case, Luis Santos has noted that there is an outlier here, and he wanted to make sure that this was not a mistake in, say, a unit conversion, but is it an actual phenotypic value. And so there's a lot of back and forth, and the shared online environment records that, and it is used by the data wranglers to approve a line for release of data for analysis. Analysis is done in a dedicated R package, a software package that applies the best statistical tests to the data. So the data may come through in different ways. A couple of centers always have controls done on the same day as the knockouts, and so in this case we do a Fisher exact. But most of the centers are doing data in batches because this is high throughput. So we have weekly batches of controls, and we have knockouts done, a knockout strain typically done over 2, 3, 4 batches. And so in this case we apply a linear mixed model that models in the batch effect that adds variance to the data. So this contributes to reproducibility, and the package is versioned and available at Bioconductor and was published a couple of years ago in PLOS One. So the point of all this is that we want to take these standardized protocols that were defined by the centers and is available in the IMPRESS resource. These protocols have been broken down into data parameters, and these data parameters are what's uploaded to the PhenoDCC and quality controlled by the data wranglers. And then once they release that data, it's analyzed in the PhenStat package. And for each one of these parameters, the whole consortium work together, biologists and developers, to associate controlled vocabulary terms to each one of these data parameters. And by this way, we can go from a knockout strain and say it has increased circulating cholesterol or decreased circulating cholesterol based on the analysis of the data backed up by this community-built protocol resource. Ontologies are also used in other places. These ontologies is another word for control vocabularies, and so we use community-developed ontologies to capture gene reporter expression using the mouse anatomy ontology, or the embryonic mouse anatomy ontology. There is histopathology done on some of the strains, and in this case we use another control vocabulary. But critical to our resource is the mammalian phenotype ontology, which is a resource developed by mouse genome informatics at Jackson Labs. It contains over about 20,000 ontology terms describing various phenotypic abnormalities observed in mouse strains. So through this whole process, we're now characterized about 3,000 strains and gotten robust phenotype data that is been released as QC data. From this, you can see on the left here, of these 3,000 lines, 60% have no previous mouse model described in literature. 40% have been described, but in many cases, these mouse models are strains that are hard to obtain because the production centers choose what models they're going to make, and they know really well what's available to the community and choose ones that are hard to find. In addition, the phenotype data that we've gathered for these 40% that have had previous mouse models published in the literature, 90% of the phenotype calls we're making have not been reported in the literature. And that's because we're doing a broad-based phenotyping instead of a very focused one. And we can see cases where we recapitulate a phenotype that's been observed in a publication, but then we add more phenotypes to that maybe the center, maybe the reporting laboratory didn't test for. On the right, we use the gene ontology as a proxy for knowledge. The gene ontology is a resource well known in informatics where curators around the world annotate gene function using a large ontology. And they classify their annotations based on whether it's experimental evidence or curated computational. Such as it has a kinase domain, so I think this gene product is a kinase. And so what we're doing is for the mouse models we have phenotype data for, almost half have no experimental annotations to them. And so now we can offer over 200 different data parameters of experimental annotation, many of which have abnormalities. So we're contributing new knowledge for the research community. We're detecting a wide array of phenotypes, and so these are broad classifications of the types of phenotypes we see. The most common is body weight. We observe this in over about a third of the strains. But you can see that we have a broad range that we detect. The gray is, by the way, is homozygotes. The blue are heterozygotes. So you can see that we're detecting phenotypes in heterozygotes where the homozygotes are embryonic lethal. One of the interesting things we're finding is we can start classifying which genes are necessary for life, and our work parallels some other nice work that's been published recently. So in in late 2015, two separate papers from the Broad and the Netherlands Cancer Institute started defining the essentialome, where they used different techniques to knock out genes in human cell lines, and just observe which ones, when the genes were non-functional, which cell lines died. And from that, they identified about 2,000 genes that are cell essential. On the other end of the spectrum is the EXACT Consortium, which collected the exomes of people who were relatively healthy. These are, in many cases, the controls for rare genetic disease studies. And what they observed is that there were over 3,000 genes that were depleted of truncating variants, meaning these genes were never seen to be non-functional, saying that these genes must be essential for life. So the IMPC is fitting into this in that we are seeing a quarter of our genes be non-viable, meaning when the gene's knocked out and they're homozygote, the embryo does not make it to birth. We also observe 10 to 11% subviability, and this is where if you remember your Punnett squares, we're breeding heterozygotes together, and we expect to have a certain percentage of knockouts, and this is subviable is when you have half of what you expect, or half or less. And so we're projecting that there might be up to 6,000 to 7,000 genes that are critical for embryonic development in mice. And we're getting a lot of interest from the developmental community and the fertility community that are trying to now associate this with human fertility and human development. So to give a couple of examples here. Here's this is KLF. This is a mouse that's embryonic lethal when it's homozygote, and the heterozygotes were phenotyped. Previous studies implicated this gene in the diaphosensory nerves, and we find that these mice are very hyperactive. You can see here on the graph on the left that we do an open field test. It's one of the first studies done on mice starting at 9 weeks of age. And you can see these mice have a significant increase in the distance traveled. And you can also see that the LAC-Z reporter shows heavy expression in the brain, but also the adrenal glands and the thyroid glands. So before this, there was no mouse model of this, and now we have a mouse model that can be investigated to study this further. Another interesting example is AHSG. This is a homozygotes. Previous studies had implicated that with a mouse knockout that there was improved insulin sensitivity, but they didn't look at the researchers didn't consider bone at the time. There was a cell culture experiment to show that this gene might be important for osteoblast culturing and inhibiting mineralization, and that there was an observation in humans that humans with choroid plexus calcification have a lower serum levels of this gene product. And so what we find is these mice have severely, very, very short bones. It's even visible on the X-ray if you look closely. And this is the type of thing we do. We can provide the X-ray images, but we also provide the center's measurements on top of that. So there's just two examples. And so how can you go find gene gerents? So at mousephenotype.org, you can come to our homepage. And we've put a lot of effort into search. The search is powered with these ontologies I mentioned earlier. And so in this case, if you look up blood glucose, you can find blood glucose in genes, which there are some genes with the word glucose in it. But in here, you can also find it in phenotypes and disease. And I have a screenshot of our phenotype tab. If you press on the tab, you can see the types of phenotypes we get. And behind that is a page where you can find lists of knockout strains associated with those phenotype terms. By far, our most popular search and page are gene pages. Most of the people coming to our resource currently are looking for information about a specific gene. So I have a couple examples of a gene page here. When you type in your gene, you go to a gene page, you get a little summary of the typical information for a gene, like IDs and names and synonyms. But then we quickly get users to a phenotype summary through icons to show you the types of phenotypes we're observing in these mice. These are based on the ontology we've been using. Below that is a table where you can actually see the granular phenotypes. Here, like we've classified things with into systems using icons. We have the actual phenotype, we have the allele, we have the zygosity. We make our genotype associations to sex. I should mention that in most assays, we're phenotyping both male and female mice, 7 and 7. And a p-value in data. And so the idea is that we bring people in, we give them an overview and let them drill down to the details. And you can go right to the data by clicking on the data graph. In this case, this is a test, ABR is a test for auditory brainstem response, where we can measure the electrical activity in the mice in response to different sounds. And so you can see here there's 6 kilohertz up to 30. And in the blue are male and female knockout mice. And you can see the threshold's much higher. And this indicates that these mice have a severe hearing deficiency. And I mentioned that we have an embryo phenotyping pipeline. And so we're starting to get in very complex imaging data from micro-CT or OPT, which are 2 different ways of measuring embryo volumes in 3-dimensional styles. So you can, if you go to our gene filter, you can filter for ones where we have some of this embryo imaging data available. And in this case, we'll take you to a dedicated tool developed by our colleagues at Harwell that gives you a Z-stack, XY Z-stacks of images that you can scan through and look at the abnormalities. And here we have an example where we have wild types and controls, and you can link them so you can scan through and you can go up and down at both the mutant and the wild types at the same time. And here we're not just saying an animal's embryonic lethal. Now we can start looking at gene function at different organ systems, and we're actually developing some automated tools to detect volumetric changes, which is very exciting because this is labor intensive currently to annotate manually. And there's also, you can look at some 3D image viewing, and if you go to an example, you can click on it and you can move it around. It's a lot of fun. So we published in Nature in September of last year about our embryo phenotyping of our first 2,000 strains. And to support that, we created an embryo landing page, and with the URLs here at the bottom. And here we give an updated summary of the list of lines we see as embryonic lethal or subviable. We give information about the procedures being used. We give you links to the different images we're collecting. We're collecting not just the 3D images, but also gross morphology and embryo laxy images. And this is proven to be one of our more popular spots in the portal, and so we're starting to build on this. And our next landing page that we're pushing up is sexual dimorphism. We have a paper in press with Nature Communications that shows the wide extent of sexual dimorphism we're detecting in mice. So in our knockouts, we can contribute about 17% of our phenotypes depend on the gender of the knockout. So we will see an increased effect in a male versus a female, or maybe we'll even get different directions. In 3.5% of our knockout mice, we'll see it up in a male and down in a female, or vice versa. This is we're very excited about this because the consortia first envisioned 5 years ago this would be important, and this is now more and more in the dialogue about how researchers need to consider both genders in their experiments, and these phenotypes are occurring in places you wouldn't expect. For instance, we do see some sexual dimorphism in hearing, which was unexpected. So one of our use cases is that we're making not just knockout strains, but these strains are models for human genetic diseases. And so we have both on the gene page and dedicated rare disease pages, we highlight this potential disease models. And we do this in 2 ways. One is kind of the orthodox way, where you have a gene associated with a human genetic disease, and then you knock out that orthologous gene in a mouse, and that's a disease model. And that's kind of the typical way many mouse geneticists think of that. But what we're doing is we're working with a consortium known as Monarch that is developed very complex scoring mechanisms that look at the phenotypes between human and mice using semantic mapping because the languages are different. And we're scoring mouse models for their phenotype overlap with the phenotype features seen in a human disease. So here's a slide provided by the Monarch Initiative Consortium, where on the left you can see here the human disease Pfeiffer syndrome that has these lists of phenotype features that are captured in a structured vocabulary known as a human phenotype ontology. And over here on the right we have a mouse model that has phenotypes using the mammalian phenotype ontology I mentioned earlier. And you can tell quickly that simple text mining is not going to match up some of these, dental crowding versus malocclusion. Text mining tool won't pick up on that. What the Monarch Consortium is doing with other developers and users of ontologies is they've generated mappings between shared anatomical features or shared genetic functions. So we've been using this. Damien Smedley at Queen Mary University, who's both a PI on IMPC and a PI with the Monarch Initiative, has run this over a data set. And we currently, in our current data release, we have over 360 new disease models with phenotype overlap. And here's a list of some here where there haven't been mouse models described before. So my favorite is this particular example. This is a FAM53. So we don't know much about this gene, but it's been detected in studies of erythrocyte maturation. It's differentially expressed during the maturation with erythrocytes and where they lose the nucleus. So what we found is that the IMPC phenotyping reveals increased hemoglobin within these blood cells and a decreased erythrocyte cell number. If you can imagine the shape, the number, and the shape and the volume of these red blood cells is critical, because if they get too big, they can get stuck in capillaries. So the size of this is very highly regulated, and you can see in the knockouts, both male and female, that these are greatly increased, and then the plasmacyte erythrocyte numbers are greatly decreased. So as I said, we don't know anything really about this gene, but now we can approach the hematology community and say, hey, we think we have a good anemia model, and open up new avenues of research and provide. And we're not just providing the data; the models are available for the researchers too, and then they can go and do functional studies on these. So this approach has proven so successful that Damien, along with the Monarch Initiative, has gone on to build tools for the clinical community. So right now, with the revolution we have in next-gen sequencing, there's efforts all around the world to sequence rare genetic disease populations that have highly penetrant alleles. So what's typically done is that a whole exome sequence will be collected on a patient and on unaffected relatives. Off-target common variants will be removed, and then in the quote-unquote traditional pipeline, a variant score is then given on the remaining alleles, ones that may impact the gene, ones that impact an active site or impact the expression of the protein. So what's amazing to me is that we just realized in the last few years that after all this, almost everyone has at least 20 different genes that they're homozygote null for, and that you can go through this whole pipeline and you can have a patient with this genetic disease, and you won't have just one variant jumping out. You'll have many, and you have to sift through that. So what Damien's done is he's created a scoring mechanism that uses the mouse model phenotype data from our resource and from other resources, from other model organism databases. And they can take those variants and then rank them based on how well their phenotypes match up with the patient phenotype. And this has really proven one of our great use cases, and is being used at a number of rare disease networks in different ways. So there's the Undiagnosed Diseases Network in the US, Care for Rare in Canada, and here in Genomics England we have a project known as the 100,000 Genomes Project that Damien's embedded in. And so our data is coming right out of the centers, being QC'd and going right to clinicians in these resources to help identify the causative variants of human disease. So one of my jobs is I want to get data out to the people, to people like you on the call, and to anyone who's interested. So we have the website, and we're always trying to refine the website, make it better. We have downloads, we have raw and analyzed data, we have spreadsheets, and I've included the URL where we have some of our spreadsheets. We have APIs, and those APIs are robust and flexible, and then through those APIs we're feeding the Monarch initiative I talked about. Mouse Genome Informatics, which is the database for mouse models for 25 years. We're using their ontologies, and so our data flies right to them. Ensemble, based here at EBI, which is used by a lot of genomic researchers. Riken in Japan has translated our website and made an RDF representation, which is a semantic representation of our data. So we've reached a whole new community in Asia through them, and there's other tools using us too. But there's I want to mention that if you remember from our pyramid slide earlier, this high production of mouse models is feeding into some pretty big secondary projects. And so I don't have time today to go into it, but there's the 3i project at immunophenotype.org that is doing a robust immunological assessment of the mice produced here in the UK, and they're actually doing separate cohorts and challenges, and that data is coming along. There's the DMDD project, which has done in-depth electron microscopy analysis of a subset of embryonic lethal mice produced by IMPC. There's the KOMP bone project based at UConn in conjunction with JAX, where they're taking breeders and they're doing a really in-depth assessment on micro-CT of bone in there, and they have a nice website with lots of interesting data. And then there's one that's bone and cartilage, which is getting off the ground here in the UK, looking at a slightly earlier time points in bone morphology in the knockouts. And then we also have a dedicated R01 where 4 different groups have been funded that are doing really in-depth embryo phenotyping some of our mice, and we have calls with them monthly to tell them about the latest. So this is just the start of the secondary projects. We think we'll be seeing more in the future, and just wanted to mention it. But most importantly, people are using the strains, they're getting the strains. So on our gene page at the bottom, you can find out what mice are available to the public and where to get them. If the mice are still being phenotyped at a center, we'll often send them right to that center. You can maybe get the mice on a collaborative basis. But this is a high throughput project, and as the mice are, after they're phenotyped, they're archived primarily at 2 repositories, the UC Davis KOMP repository, and which all the mice generated by NIH funding goes there. And then there's the InfraFrontier resource here in Europe, which mice produced in Europe for the IMPC project are archived there. So these strains are available for you. They're sitting there in archives, and we know they're being used because we've just started really analyzing this. People are citing us at a quite astounding rate. So in this top graph, these are publications that have cited using mice data or embryonic stem cells over the past years, and we're just near 2,000. I checked yesterday, we're at 1,997. That means we'll be at 2,000 soon. And this is using curation done at MGI and combined with some with curation done on my team in conjunction with text mining. And at the bottom here, you can see, that the grants that are citing this are quite astounding. So we have over 200 grants from the NCI, the National Cancer Institute, that are using our mice, and you can see on down the list that we are paying back to the research community in a quite astounding way by making mouse models that are of high quality with data that you can trust, that can start generating hypotheses for other researchers and to investigate them. So my conclusions are high throughput phenotyping in the IMPC is revealing thousands of new gene phenotype associations. The data is all freely available, so please take a look. These new mouse strains are being deposited in public repositories where researchers can get it for a low cost, and the project is ongoing. Just to show that we're now initiating a late onset phenotyping pipeline. As we enter our second 5-year phase, we are aging about 15% of the mice funded by the NIH are going to be aged for a year and looked to reassessed for phenotypes to look at how to look for phenotypes that come on at later onset. This is also giving us exciting opportunities to do non-invasive testing during the months in between. And so we're actually having a meeting next week to discuss this and other issues and how we can generate more and better data for the research community. And this, of course, I get to represent a tremendous group of people, and here are the list of the partners in the IMPC. I don't have a listing of all the funding, but the informatics is funded by the NIH Common Funds for the funding mechanism you can see down there. And so thank you very much for coming, and I hope you enjoy looking at our resource.